So when you think of Meiosis one is where all the genetic variation is occurring. We've got the homologous chromosomes pairing up, called synapsis. Oh, we'll highlight that word. We have the formation of those two homologous chromosomes. We call that a bivalent or a tetrad. Same name, same thing. And then that independent assortment is when those bivalents or tetrads line up on the middle of the cell. That is another source of genetic variation. So prophase 1 and metaphase 1 is where all the action is in getting that genetic variation. And then one other thing happens in prophase 1, and that's when they exchange those little pieces of genetic information, and we call that what again? When the sister chromatids share a little bit of each other? Crossing over. So that's not listed here, but that happens as well. And it's really hard for me to write with my mouse, but you can, yeah, I can't figure out how to do that. Where? Here? Highlight what word? Okay. Oops. Okay. Amazing. Crossing over. Oh, but see, but then you can't see it on there. That's the only bad part. Yeah, not on a PDF. That's the, yeah. I know, I wish there was a better way. Okay, so anaphase then, we pull these apart. So we're basically just pulling apart those bivalents. So we separate, but the sister chromatids do not separate. And that's a key thing to remember is Meiosis 1 is all about those tetrads. We're not pulling apart sister chromatids yet. We're not separating the X chromosome yet, you know, the X-shaped chromosome. We're just separating the homologous chromosomes. And then we have this short period where we go between Meiosis 1 and Meiosis 2, and that is called interkinesis. A key thing that happens there, I'm going to highlight it in pink, is DNA replication does not occur because we already have the sister chromatid, right? We already have chromosomes that look like this with the centromere. We already formed that before prophase 1 started. All we did was separate those homologous chromosomes in Meiosis 1. Now we're going to separate the sister chromatids. So Meiosis 2 looks just like mitosis. It's very much like mitosis. So if you know mitosis from our last unit, then Meiosis 2 is just the same processes. Nothing special is happening in there. We're just running through the phases, prophase, metaphase, anaphase, telophase, just like mitosis, and we're separating sister chromatids. Very similar, Meiosis 2 and mitosis, nothing special, just condensing chromosomes, lining them up, pulling those sister chromatids apart now. So this is where we do pull them apart. No. Yep, just Meiosis 1. Yeah, Meiosis 1 is where all the genetic information is being varied a little bit. So we have some variety in these 4 cells that are produced, right? But Meiosis 2 is pretty boring. It's just, okay, we did all the action in Meiosis 1. Now let's separate those sister chromatids and make 2 more cells each. So we have 4 total. So this is where all the action occurs. We got crossing over, synapsis, independent assortment. That's all Meiosis 1. Meiosis 2, pretty boring. So I'm going to make this bigger so we can see. So prophase 1, here we can see they come together. There's the 2 homologous chromosomes, the red one and the blue one. So this isn't, you know, in the egg and testes of the male, right? In the male throughout life, and the egg of the, this is formation, this is the ovary, I should say. This is the ovary of the female, and she's still in her mother's womb. And I was telling your story over the weekend about someone saying that, was it Victoria, did you say about we're actually in our grandma, the egg that formed us was formed in our grandmothers, right? Yeah. And I'm like, wow, that's really deep, and that is very true. And Nick, I think you elaborated on it because I wasn't quite understanding it, so you guys, yeah. And it's very true. So this is in the ovary of the female before birth that this process is occurring, right? So we have this 2N cell in the ovary that enters into meiosis and is going to form eggs that will later be ovulated after puberty in that female and be fertilized. So this is the chromosome from grandma and grandpa, right, that are coming together, those homologous chromosomes. So there's the tetrad or the bivalent, right? And then they line up. So these are the bivalents. They separated them already here a little bit at metaphase 1, but they were connected by synapsis. You can see the little bits here. This is red and this is blue because crossing over occurred here, lining up, being pulled apart. Then again, we jump right to this interkinesis. So they're, telophase 1, everything kind of reforms, but it doesn't recoil and become chromatin again. That would be a waste of time because we're not done with these cells. We need to divide them again. So they're going to enter into, and notice that the chromosome number is half now. So we went from 4 chromosomes in metaphase 1 at the start to now we have 2 chromosomes. So all the action again is meiosis 1. If you can just remember the 4 steps of meiosis 1, meiosis 2 is just exactly like mitosis. So we've got 2 chromosomes out now. Now each of these is going to divide one more time. So if this divides into 2 and this divides into 2, we have a total of 4 cells, right? So now the job of meiosis 2 is just to pull these sister chromatids apart. So that's what happens. So here's each of those cells, separating the chromatids in anaphase 2. And then telophase 2 is breaking them apart, and now we've got 4 cells. And you can tell the difference because they're no longer sister chromatids, right? They're individual cells. So for these cells to move on and make more cells, if this was mitosis, we'd have to undergo DNA replication to get the X back, right? But we're not going to do that because we want sperm and egg to be haploid, to have the N equals 2, that's chromosome number. So it has 2 chromosomes. We want these to be half the number. So if this is an egg, when that egg is fertilized by sperm, which is also half, now we're back to the double set again, and you've got a zygote, and mitosis takes over from there. So we have 4 genetically distinct cells. Because of that crossing over and independent assortment, you can see that each cell here at the end of meiosis is not the same as the others, right? They're all a little different. And we talked about how amazing it is that brothers and sisters look so similar when there's such genetic variation among sperm and egg. Okay, so this is just a comparison chart, but I gave you that handout too in Blackboard that you can look at that describes the differences between the two. But it's all about genetic variation. That's the goal of meiosis, and we need it to make sperm and egg. So mitosis is just a copy machine. Everything's genetically the same. So what I would recommend is that you take this great little diagram here, this meiosis 1 and meiosis 2, and be able to distinguish among these cells if they were separated from each other. Could you put them in the right order if they were all scrambled up? So you could print those out and cut them up and try to rearrange them. So when we look at how plants exist in nature, they aren't like us. We're all diploid, right? Everybody in this room has cells in your body outside of ovary and testes, the sperm and egg. Everybody is mostly 2N cells that were formed by mitosis. You started out as a zygote that had 2 sets of chromosomes, and then copy machine is everything else, right? And that's why we use that for, DNA fingerprinting. We can find out who did a crime by finding sloughed off skin cells and things that are left at the scene. But plants have forms out in the woods that are haploid. They actually have versions of their life cycle that are haploid, and we call those gametophytes. Gamete, right? So you can kind of see the prefix in there. So a gametophyte has only 1 set of chromosomes. So when you say it's haploid, It has one set of chromosomes. And when it gets a little nicer out, you can look for these versions. I'm just going to abbreviate that, out in the woods, and I'll show you some pictures. And then the diploid version is called the Sporophyte. And the Sporophyte is larger and more obviously seen out in nature. So this is 2 sets. So we say 2N, and this would be N, 1 set of chromosomes. So let's take a look. I want to show you a picture of that. Gametophyte. So if I go hiking in the woods and you see a moss growing on a tree or on a rock, if you look real closely, you can impress everybody, and you see these, you'll see these thin little spindly stalks coming up from that moss. Most of the moss you see is the furry carpet looking stuff, right? That's the Gametophyte version of that moss. So these are N individuals. They have half the number of chromosomes. And then the Sporophyte is the 2N version of that plant. So there's 2 different life cycles of these, and there's female and male gametophytes. Yes?
It's kind of like when you let the grass grow too long and then it grows like the seeds kind of on the top, is that type of thing?
I'm not sure.
When you let grass grow too long,
I don't know. We could look that up. I want to show you another version of fern gametophytes. These are hard to find. And when I taught biology over at UWL, we grew fern gametophytes because they had like a greenhouse over there, and it's kind of cool. And these male gametophytes actually produce sperm, and we looked at it in the microscope, and it looks like human sperm. They have little tails, and they swim to the female gametophyte, and then a sporophyte grows from that. I'll show you what that looks like. So, here. So the gametophyte is a little heart-shaped, leaf-like structure really close to the ground. So if you look at a bunch of ferns and you get down real close in the ground, you'll see these small little heart-shaped structures, and they produce sperm and egg, and they eventually will grow up to form the sporophyte, which is what you see. So I'll show you a real one. Let's see. These are all really tiny. Here. See these tiny little heart-shaped structures real close to the ground? Those are the gametophyte version of a fern. And then the big leafy part is the sporophyte generation. So if you take a course in plant biology, you'll learn more about that whole process. But it's just worth mentioning here that animals are different than plants, where animals only exist as the haploid version only exists in sperm and egg, the single cells, right? Once they come together, we have the diploid version, and that's it when it comes to animals. But plants have 2 different forms of life. They have the gametophyte and the sporophyte. Okay, so throughout the life cycle, yeah, especially when you look at mosses, we see them throughout. I'm going to look and see. I'm going to pause this for just a minute. All right, so animals, like I said, they are diploid. They don't have an adult form that's haploid. The only thing we have are haploid gametes, which are the sperm and egg. So when we make gametes, it's meiosis. So it's the same thing. When we say gametogenesis, we're talking about the process of meiosis, making sperm and egg. But there's a little slight difference in how sperm are made and how eggs are made in terms of the cells that we get at the end of the process. So if we look at We talked about sperm and egg have one set of chromosomes, and when they come together, we have two sets again, and then we call that a zygote, and that undergoes mitosis just to make a baby, right? So if we look at the process of meiosis, again, we're looking at meiosis 1 and meiosis 2, what do we end up with? We end up with 4 cells that are genetically distinct from each other. But in the female, it's a little different because the egg, when it's undergoing meiosis, it's stuck in metaphase 2. It's stuck there until fertilization takes place. So the signal for it to continue meiosis 2 doesn't occur until that egg is fertilized by sperm. So once the egg is fertilized by sperm, then those final chromosome separation occurs, Anaphase 2, and sometimes problems happen with that. If chromosomes don't migrate like they should, like in an older egg, the spindle breaks. People think the spindle gets brittle. It breaks, it dumps off an extra chromosome in one of the cells, and if that's the cell that the sperm nuclei fuses with, you have an embryo or a zygote with a chromosomal anomaly or wrong number of chromosomes. And oftentimes, the wrong number of chromosomes, if it's one of the larger chromosomes, like 1 through 18, those babies don't always survive. And people can have multiple miscarriages because of problems with this Anaphase 2 not happening correctly. So that's a unique thing that happens in females. Another thing is when the cells are undergoing meiosis 1, meiosis 2, some of these cells will, I'm going to try to see if I can find a better picture. They don't divide evenly. So when that ovum divides in meiosis 2, it's not an even division. I didn't spell it right. There we go. So we get something called polar bodies. So if we look at, this is a good example. So if I look at, so we have meiosis 1. So it's called the primary oocyte. It divides into 2 cells at the end of telophase 2, but it's not an equal division. We get one cell that gets a lot of the cytoplasm and the organelles necessary for development and to continue on, and then we get this tiny little cell called a polar body. And a polar body doesn't have enough genetic information, doesn't have enough cytoplasm, and it dies off. So we really have, we're supposed to have 2 cells at the end of meiosis 2. We really, in females, only have 1 because of this polar body is a non-functional cell. And then in meiosis 2, when each of these cells divide again, this cell might divide again, and you might have 2 polar bodies, and then this cell divides again to become, again, only if fertilized does it complete meiosis 2. We have the zygote and a polar body again. So at the end of meiosis, we have, in females, we actually have 4 cells. So we have the first polar body 1, and then we have this cell, which is the ovum, and then this might divide again to form 2 more. So we have 4 cells, but they're smaller. So if I can put this in a, just make it a little easier. So we start out with, trying to think of the best way to do this. Insert a shape. Where's my shapes? Right here. Okay, so you have the 1 cell, and then it divides by meiosis 1 to make another cell, right? But the second cell, like I said, is what? A polar body, right? So I'm going to move these over. So this polar body is really tiny and insignificant. Looks like that. Then we go through meiosis 2. This cell is going to divide again. This is the egg again. So we're going to have 2 new cells. Again, an unequal division. So we got a tiny little polar body again. And then this may have enough genetic information, not always. It may divide again in that meiosis 2. to form 2 more polar bodies. So meiosis 1 is occurring here. I'll just slide these over so they're more even. And then this is here, this is here. How many cells do I have at the end of meiosis 2 if fertilization took place? 4, right? And that's what meiosis 2 tells us, is we should have 4 cells at the end. Hold your thoughts for 1 minute. But if we look at sperm, we need lots of sperm to find that egg, right? So it's more advantageous for us to make lots of sperm. So we're going to have 4 cells with equal divisions when we talk about spermatogenesis. But with oogenesis, we have unequal divisions, because how many eggs do humans want to produce? 1, right? So 1 healthy, viable egg for 1 fetus. But lots of sperm available to find that 1 precious egg. So in like cows, for example, they usually have 1, maybe 2, just like humans have 2 sometimes also, right? But they are set up for 1. They ovulate 1 egg. So what happens in pigs that have 8 litters of 8, or dogs that have litters of 8? In 1 cycle of popping out an egg at ovulation, they're going to put out more than 1. There's going to be 8 of these. And this process of forming 1 egg and 4 polar bodies is going to happen with all 8 of those eggs. So a pig or a dog or a cat is going to do this, but it's going to ovulate 8 of these, which are all going to produce a bunch of polar bodies and 1 viable egg for fertilization. Okay, question? Victoria, you had a question? The ovum is the name of this cell. When we're down, this is called an ovum. So this happens in the ovary. It's called a spermatozoa, the final version of the sperm. Is that what you're asking?
But like this cell, like if we have the ovum that like holds the egg until then
It's not, it's called an ovum only after it's been fertilized. Actually, this is called a secondary oocyte, this cell here. And the secondary oocyte is ovulated. And then when sperm fertilizes it and it finishes meiosis 2 and we get this final polar body division, this is called an ovum. Oocyte. And I'm not going to hold you to that kind of detail because it's not a human anatomy class, because plants, you know, are different. So they have different names in plants. But so you can see here, primary oocyte, primary spermatocyte. At the end of meiosis 1, we have secondary spermatocytes, secondary oocyte. This is what is ovulated. Can you see how the sperm comes in here? So we're stuck, like I say, in metaphase 2, and we complete that after sperm fertilize. And then we have a zygote. Yes? None whatsoever. They're just wasted space. Yeah, yep. And again, it's just because we don't want to lose nutrients and cytoplasm to cells that we don't need all those eggs. And even the number of eggs that are formed by meiosis in fetal development, a bunch of those, like they're like half in number. I don't know the exact numbers off the top of my head, but the number goes down significantly even before birth. A lot of those will atrophy and that become non-functional. And then throughout adolescence, there's more selective destruction that happens of these eggs. So we only ovulate about 300 over a fertile lifespan for females. So there's like, I don't even remember the numbers. I'd have to look. I don't have my anatomy book with me, but the very large numbers. I mean, I can look it up. Just how many eggs are formed in human ovaries. Get some quick numbers here. Okay, so you're born with 2 million eggs, 11,000 die every month prior to puberty, and only 300,000 to 400,000 remain. And then another 1,000 die each month. So it's kind of a depressing process, isn't it? But we're all here because of a surviving egg in our parents, right? So it works, right? But we have
Did the math, like every egg for every possibility.
right, exactly. Yeah. So it controls our population, doesn't it? Okay. So back to this then. So what happens when we get the wrong number of chromosomes? Like I said, when sperm fertilizes egg and we're spreading those sister chromatids apart and something breaks and dumps off an extra chromosome, we have chromosomal anomalies. So monosomy means you only have one of a chromosome. Trisomy means you have three of a chromosome. And trisomy, a real popular trisomy condition that occurs in humans is Down syndrome. If that's a disorder, due to chromosomal issues with chromosome number 21, extra pieces of chromosome number 21 or entire whole extra chromosome in its entirety ends up in that final egg that is fertilized. Trisomy, yep, an extra, because we're supposed to have two of each chromosome, right? But instead, a person has three. So then you see the effects of that in chromosome. So the information on chromosome 21, when we have extra information that's carried on chromosome 21, you see the characteristic features of people with Down syndrome. And that is a very small nose and very little bridge to the nose, a small oral cavity and a large tongue. So if you look at people with Down syndrome, oftentimes they have trouble speaking because they have a really small mouth and a very large tongue. Sometimes they hang their mouth open to breathe because of the small space and the large tongue. A lot of heart conditions, a lot of digestive problems, increased risk for cancers, cognitive delays that vary quite a bit. Some are very high functioning and hold jobs. Some are very low functioning and need a lot of support. Oftentimes, they have really short stature, metabolic problems. They have trouble with weight gain, that they don't metabolize foods appropriately, and they're often overweight. So when you look at people that are like that, it's just the result of that extra chromosome 21. It just kind of throws so many different body systems off. Hearing problems, vision problems. And then another thing they look at birth, which is like an instant sign, is on the inside of the palm. Look at the inside of your palm. You have a crease on the inside of your palm. It looks like an M. Can you see an M on the inside of your palm? I'll show you a picture online. M palm crease. Isn't this great that we have the internet at our fingertips like this? When I was in college, it was not that way. Do you see the M? It's kind of at a sideline. Okay, that's a classic characteristic that you do not have an extra chromosome 21. But yeah. So single. Now, this does not mean you have Down syndrome if you have a single palm crease, so I don't want you to go home thinking, oh my gosh. But people with Down syndrome have the single palm crease. That's a characteristic of Down syndrome. So can you see the single palm crease that comes across? There's no M. See that? Just a single palm crease. So that's a characteristic. And when my daughter was born, I was 41 years old. And I was watching her, they took over to the little incubator, and the first thing they did, because everybody thinks, oh, old mom, chromosomal problems. And, yes, the risk is higher. It's 1 in 100 versus 1 in 10,000 of a 20-year-old, but it's still 1 in 100. So I wasn't, mortified during my pregnancy. I kept the statistics in my mind that chances are good she's going to be fine. And I did the testing, not the invasive testing, but just the little blood test they give you and the ultrasound. They look for a lot of things there. Everything looked good. So I thought, we're fine. Anyway, the minute they put her on the incubator, I saw them take both of her hands and open them up and look for the palm crease. And I'm like, oh, I know what you're doing. Whatever. That's fine, because you'd want to know, right? And they were looking for that single palm crease. Anybody have the single palm crease? It's okay. Like I said, people who do not have Down syndrome may have a single palm crease. No? Everybody has the M? Just for trivia's sake, you know that tabby cats have an M, right? You can look for it now. If you have a true tabby, see the M on their forehead? See right there? That's the tabby cat. You know, you can look for the M. Because of the M, so you notice that, yeah. All right, so trisomy. So here's chromosome number 21. There's 3 sets, or not 3 sets, 3 copies of that chromosome. So I have read, I haven't done the research on it, but I have read that children with Down syndrome are more often aborted by people that are overwhelmed by that diagnosis and don't want a child with Down syndrome. And they also end up in, you know, being given up for adoption because people can't handle the, you know, the extra care that people with Down syndrome need. But also people that know, that understand what Down syndrome is, love children with that condition, and they get adopted very quickly because they have a childlike, many of them, because of a mild cognitive delay, they have a childlike view of the world, and they're very happy people, and they love music. And people say that people that have had a child with Down syndrome will say it was, you know, the best thing ever, and they're like a celebrity in the neighborhood, and the kids all love their brother or sister that has Down syndrome, and they're wonderful people. So when we think of genetic anomalies, it's not always terrible. But for me, I wanted to know, so if I had a child that had Down syndrome, I could be prepared, right? You want to make sure you're prepared for, because sometimes they have feeding issues because of the oral cavity, you know, size of it and the tongue control. They have trouble feeding. So you want to be prepared, and just knowing that, you know, Special Olympics and different jobs in the community, there's people that have Down syndrome that love their jobs, and their jobs are just basic jobs that don't require a lot of skill, but they just love what they do, and they really make other people's lives a brighter place. So I want to put that little piece in, that it's not always a terrible thing if someone has Down syndrome, because they are very happy people. Okay, Turner syndrome. This is another condition. This is a monosomy where you have a single X chromosome. So again, if that spindle breaks and that X chromosome doesn't go to the cell that's fertilized, they only have one X chromosome. So that means either the sperm or the egg was missing an X when it was formed. So when sperm and egg came together, there's no X, right? So as a result of that, a person has Turner syndrome. And Turner syndrome causes infertility. It causes short stature. It can cause sometimes mild cognitive delays. So people might just be a little bit learning disabled, or, you know, just not of average intelligence, but nothing that you would really notice. Because I've known people that have Turner syndrome, and oftentimes they have large moles on their face, and what else? Like I said, infertility is a big problem, though, because I didn't know that. My daughter went to school with a girl that had Turner syndrome, and she just loved my babies, because my daughter was, when my kids were in grade school, my oldest, so I was having babies while my kids were in grade school. Everybody wanted to pass the babies around at games, at basketball games and stuff, and she was always wanting to hold my son. And I kept telling her mother, oh, she's going to be a great mom one day. Oh, she just loves babies, and oh, she's going to be a great mom. And then later on, I found out that she had Turner syndrome. So she could still adopt, of course, right? But she is a little bit delayed, but not very much. But she does have certain characteristics that you can tell she has Turner syndrome. Klinefelter syndrome is, what would this be an example of? How many copies do we have here? It's a trisomy, because we have 3 copies. So this is an extra X, so you have a boy with an extra X. And because of that extra X, it causes some changes, which is really long arms and legs, large hands. Normal intelligence in most cases, maybe a slight learning disability. Some people have said, and I haven't researched it, is, what's his name, the swimmer, Michael Phelps? They say has Klinefelter syndrome.
I think a lot of athletes have long arms, long legs, and large hands.
Yeah, yeah. So, yeah, I don't know. But anyway, so again, that interferes with fertility as well because of that. Oh, yeah, right. Yeah, that is often chromosomal, like people that are hermaphrodite, where they have female parts, but something develops and there's a small penis that develops at birth. Yeah, that is definitely a mutation and related to chromosome. But I don't know what the name of that is. I'd have to look that up. But yeah, because the X chromosome carries information not only related to female gender, but also other structural, features of the human development. But the Y chromosome, look at how small it is. It has primarily just directions for making a baby a male. So most of these other, chromosomal problems we see that happen at development or fertilization are related to the X, because there's so much information on the X that that's above and beyond just female gender. So because of that, more males are miscarried and have problems, because if girls have a good X and inherit a bad X, they can sometimes survive and be just fine. But if males develop a bad X, they don't have a good X. All they have is the Y for the other sex chromosome that makes them male. And those problems with the X can be fatal and cause miscarriage. So there's a 50-50 chance of a male or female to be formed at fertilization, but more female babies make it to birth than male babies because of problems with the X chromosome in male babies. So all these little problems that can happen in chromosome structure. We can have deletion, where you have pieces of genetic information that are missing, or a duplication, where you have, when DNA is replicating, you can add an extra piece of DNA in that process. So DNA polymerase, we talked about that, right? During DNA replication, when it's copying that strand, it's semi-conservative. Sometimes extra pieces of DNA are added, and we call that a duplication, making a longer chromosome. Translocation is when we have a segment from one chromosome moving to another one that are not homologous. So there's a lot of different things that can happen among these chromosomes. And inversion is when it flips in an opposite direction. And then again, this is going to interfere because this is DNA, right? Chromosomes are DNA, which code for proteins, which code for structure and function in the body. So as a result of that, you're going to have problems with function or structure. And we see a lot of that. So we're going to stop here, and we're going to look at meiosis in a little bit more detail with some of the handouts I've provided in Blackboard and those animations before moving on.